So I'm sorry we got cut off before, but what we were talking about was metabolism and respiration in prokaryotic cells and in eukaryotic cells. And they all need to use energy efficiently. They all need to take in nutrients, process those nutrients, and process them into energy. That is done by a set of organelles in the eukaryotes that start with the vacuoles, which can store nutrients and water that can be taken in by mitochondria in order to break down the nutrients into ATP, and then for a respiratory process to move that ATP to ADP to produce energy. So we think of a respiratory process in our situation as taking in oxygen and releasing carbon dioxide. And in taking in oxygen, our cells are using that to break down ATP into ADP so that we have energy. And the waste of that is carbon dioxide. In eukaryotes, you can have that combination of vacuoles and mitochondria for eukaryotes, especially in the Animalia kingdom, that need to take in nutrients. And in the plant kingdoms, you can have chloroplasts instead. Chloroplasts in plant eukaryotes act as a metabolizer and as a respiratory site that uses the energy of the sun to produce energy. So it's interesting that chloroplasts can actually be anaerobic and aerobic and can function as an energy source for the cell outside of nutrients that come into the cell. So that's for eukaryotes and some of the organelles that are involved with the metabolic and respiratory processes. There are other organelles that are involved with eukaryotes, and some of those that are important and that you will need for your activity will be Golgi apparatus and endoplasmic reticulum. And both of those are involved with proteins. They're involved with moving those proteins to other areas of this big cell, relatively big cell, that a eukaryote is. Again, the eukaryote is about 50 times larger than a prokaryotic cell, so that there is some packaging of proteins involved and some finishing off of proteins and processing of proteins before they are moved elsewhere in the cell. Interestingly enough, the Golgi apparatus, I was wondering where Golgi apparatuses are in abundance in what types of cells, and they are actually in abundance in goblet cells, which are cells that produce mucus. And I thought about that, and those are actually some of the cells that are most debilitated by COVID. COVID seems to really nail the goblet cells in your nose and destroy some of their mucosal activity, which mucosal activity attracts a virus to expel it. So that, as you can see, would really cause an infection to speed up and accelerate. If you want to remember that about Golgi apparatus and what they do, they are secretorial cells that process a lot of protein, mucus, and one mnemonic for that might be Golgi goblet cells make gunk. Okay, so if that helps you out, those are secretorial cells that do produce an enormous amount of protein, and they are in eukaryotic cells. I wanted to also point out to you that another thing that prokaryotes and eukaryotes have in common are a cytoskeleton. And a cytoskeleton adds shape to a cell, and you're going to need to pick out some shapes in your activity. You have 3 shapes up here for your prokaryotes, but you have more shapes for your eukaryotes. your eukaryotes can use any one of these three shapes that were for the prokaryotes, but then there are others for eukaryotes, including one that looks like a squamous shape to me, a cuboidal shape, a cylindrical shape, and a fusiform or an ovoid shape. And this is a amoeboid shape, and like a polygonal shape as well, an amoeboid shape. And so the cytoskeleton for prokaryotes and for eukaryotes is very similar. It was certainly moved down by evolution, and it consists of different fibers and microtubules and microfibers that exist for both prokaryotes and eukaryotes. And those microtubules are not only inside the cell, but versions of them become flagella, which are the locomotion means for prokaryotic cells and eukaryotic cells. It's one of the locomotion options is to have a whipping flagella microtubule that propels the bacterial cell or the eukaryote or the protist cell through medium. And this is a graphic in terms of a prokaryotic cell and its flagella, and it is just spectacular to see this under transmission electron microscopy. Look at this beautiful design that looks absolutely like a stage for a whipping flagella that can propel a bacteria or a protist into a medium. In terms of the prokaryotic flagellum, this is a model from E. coli, and E. coli is one of the bacteria that can cause a virulent urinary tract infection in the urethra by just climbing up the urethra with its rotational movement. And this is known, this base here is known as a motor it's a proton pump motor that is similar to the microtubules that really hold the shape of the prokaryote that it's from. Now, flagella for prokaryotes are different from flagella in eukaryotes. They apparently evolved differently, but obviously there was a need for this locomotion, and they evolved in very similar ways with slightly different materials. So a flagella could be a single flagella, and you see it in sperm as eukaryotes, but flagella are very well known in the prokaryotic world as well. And I want to show you that you have a choice in your lab to choose from one flagellum or polar flagella on each side of the prokaryote or the eukaryote, or multiflagella. And in terms of eukaryotes, you have more options besides flagella. You have options for cilia, and you have options for pseudopods. So for cilia are coordinated hairs that can cover a eukaryote like a protist, and instead of having one or two or multiple flagella, you could have hundreds or thousands of these coordinated hairs that move together about once a second to propel the protist in the direction that it wants. So you could see here that the cilia are on the perimeter of a eukaryotic cell, and that might lend itself to a shape that is more spherical, like a spheroidal cell, or it might be something that's better with a rod-shaped eukaryotic cell, or maybe even an ovoid shape as well. So something that has a large surface area and that might have a directionality to it, or has a circular motion so that all of these cilia could be coordinated together. It might be harder to do cilia effectively on other types of cells. So keep that in mind when you put together your eukaryote. And I also wanted to point out to you that for amoeboid cells, their options include pseudopods, which are extensions of the cell membrane to propel yourself forward, almost like putting out a little feeler and moving it forward. So pseudopods can extend out the cell membrane, but they're only linked with amoeboid shapes. I hope that helps you a bit with the cytoskeleton that is similar and related in both prokaryotes and eukaryotes. I'll see you on the other side.